So let's read the last statement first. The colony characteristics of the microbe involved are except. So basically you'll have to identify the microbe and you should have the knowledge of the colony characteristics of that particular microbe, and then you have to find out the exception. So let's start with the question here. A 6-year-old boy develops a mild fever and sore throat. Now this sore throat is something which is very special. On general examination, body temperature is 38.3 degrees that is Celsius, and tachycardia is generally seen. Nasal discharge is also present. Now if you culture the discharge, so culture of the discharge is done to detect the microbe. So basically this is the culture of the nasal discharge. Okay. Now here, if you just try to understand here, you will be able to see that there are certain club-shaped bacteria. These are the club-shaped bacteria. These are either seen in pairs, they can be seen in pairs, they can be seen in clusters or patches. So you will be able to understand here, they might be seen in pairs somewhere, and most of the time they will be seen in the patches or the clusters. Okay. Now another very important thing is that they will be giving a characteristic pattern that is known as the Chinese letter pattern. So if you see Chinese letter pattern, this will be another characteristic feature of these bacteria. So from all this information, we come to a conclusion that this is nothing but your Corynebacterium diphtheria. Okay. So basically it is Corynebacterium. Now if you just talk in details about the Corynebacterium, so according to McLeod's classification, there are 3 types of Corynebacterium diphtheria which can be considered. Okay, he classified those into 3: C. gravis, C. intermedius, and C. mitis. So first of all, we'll talk about the colony characteristics of these diphtheria. So if you just try to grow these, incubate these bacteria on the selective medium, that is your cysteine tellurite blood agar. So what is the selective medium for these? Very important question again. Selective medium will be cysteine tellurite blood agar, and they generally grow in 2 days. Okay. Another important thing, if you just want to do the rapid growth, so if you want to do the rapid growth along with the Mycobacterium tuberculosis, these bacteria, Corynebacterium, are generally grown on the Loeffler's serum slope. Okay, so we will be using Loeffler's serum slope for growth of rapid growth, that means 6 to 8 hours growth of these bacteria, that is Corynebacterium. At the same time, we are using this particular agar for this particular media, that is Loeffler's serum slope for growth of Mycobacterium tuberculosis, right? So these are certain important facts which are asked in exam. So let's start with the colony characteristic. The colony characteristic on tellurite will be, it will be showing daisy head appearance. Daisy head appearance is shown by gravis. Frog's egg colony is shown by C. intermedius, and posed egg colony is seen by C. mitis. Okay. Then the second important feature which we have to consider here is hemolysis. So if we talk about the hemolysis, hemolysis is positive in C. gravis. It is non-hemolytic in case of intermedius, and these are usually hemolytic in case of C. mitis. Here I should make a correction here. C. gravis, it may show, may not show. It can be variable, not positive. It can be variable. So again, I'll repeat this. Hemolysis will be variable in C. gravis, non-hemolytic in C. intermedius, and C. mitis will be usually hemolytic. Then if we discuss about the glycogen and starch fermentation, glycogen and starch fermentation, that is positive for your C. gravis, negative for your intermedius, and again negative for C. mitis. Then we'll see the most common complication associated with the infection of these bacteria. So most common complication which is seen when the person gets infected is paralysis or paralytic and hemorrhagic. That is with C. gravis. C. intermedius, hemorrhagic. And finally, if we talk about the C. mitis, they will be very life-threatening obstructive lesion. Obstructive lesions are seen in air passage. Okay, so they can obstruct the air passage basically. So these are the various important features which we have to remember about the culture of the Corynebacterium, and it was a classification given by Macleod. It is according to the Macleod's classification. So if we just go back to the question again, and we will be able to identify, yes, these are the Corynebacterium, and at the same time we have three types of Coronones, that are frog's egg, poached egg, and daisy head. So daisy head with C. gravis, poached egg with C. mitis, and frog's egg is with C. intermedius. So medusa head is not seen in the Corynebacterium diphtheria. So answer to this question will be 4, that is medusa head. So if you want to read the explanation, you can go at the bottom and you can read more explanation attached to it.